As a myeloma doctor, Dr. Banerjee uses examples of real-world data from multiple myeloma. However, this information on real-world data can apply to all blood cancers. Why may real-world data not always reflect what was learned in a clinical trial?
Patients who enroll in clinical trials, I am tremendously appreciative of their time and effort. I joke to patients they're putting their blood, soil, and tears into, this clinical trial for the betterment of myeloma care for all future patients, but they're not always representative of some of the other patients that I see. So I think that's the biggest difference. So two things can happen. One, patients who have the wherewithal to be at an academic center, at a major trial center where they're able to consider a clinical trial, are often healthier, have more resources, have more health literacy. Clinical trials, even though we reimburse patients, they often cost money for the logistics of getting to the academic center and are not always reimbursed properly. So it's tricky. Being a clinical trial is not easy. As I alluded to earlier, clinical trials involve a lot more time of the patient, unfortunately. Many more tests like ultrasounds and echoes and bone marrow biopsies, often more time in clinic. As I alluded to, clinical trials often use older styles, for example, requiring a 24-hour urine assessment every 3 months. That's extra time stuck urinating into a jug and bringing it back to clinic. That's not for everybody. So I think that's one part of it. It's the patients whom we approach for clinical trials or even approach our academic center for clinical trials are different. And the other part of it, and that's not, I can't easily fix that. I wish that I could. The other part of it that we are trying to fix as a field is that clinical trial protocols are often restrictive. Clinical trial sponsors, meaning whoever is paying for the trial to be run, sometimes often drug companies, not always, sometimes we have IITs, which are investigator-initiated trials, meaning this my idea, I'm using my research funding to pay for this. In all of those cases, however, we want to choose patients, we want to be able to see whether the drug is safe, whether the drug is effective. And unfortunately, patients who don't have the lab parameters to easily be able to prove either or have issues with either lab parameters, their blood work to prove safety or prove efficacy, often are not allowed onto those trials. What do I mean by that? For example, we want to make sure that this new drug doesn't make people's blood counts get too low. And so we have good ways to assess for that, but that requires the patient going on to the trial have good blood work to begin with. So patients who have cytopenias, meaning low blood counts, often are not eligible for clinical trials, which is unfortunate. Or efficacy. We want to make sure the drug is actually working, making the M spike go down, making the plasmacytomas shrink, making the kappa and lambda ratio more normal or something along those lines. Patients who have oligosecretory or non-secretory myeloma, meaning they don't have those blood parameters that we can easily track, are all of a sudden excluded from these clinical trials, and it's unfortunate. So I think that's, between those two, that explains a lot of why trials and real-world data often reach different conclusions, just because clinical trials, the patients are healthier in a lot of ways. And so a good example of this would be with teclistamab or elranatamab. So teclistamab is a good example. It's a BCMA bispecific antibody. And the Majestic-1 study, we saw very impressive durations of responses, a duration of response of 18 months. So patients where that drug worked, it worked for a good solid year and a half on average. And it worked in over 60, 70% of patients. Several real-world data sets have emerged since then. I've looked at this in real-world patients receiving teclistamab, also known as Tecvayli commercially. And it's encouraging actually to see that the duration, the depth of response is the same. The response rate is the same. Still, about 2/3 of patients, maybe 50 to 60% of patients can expect to see a clinical benefit from teclistamab. But for whatever reason, compared to the trials, we have not really seen the same duration of response in the real-world setting, where the drug doesn't work for as long. Why is that? Difficult to say. My guess is that real-world patients, in the real world on these clinical trials, their T cells aren't as healthy. T cells need to work for teclistamab to work. In real life, teclistamab doses are often held for infections or complications, or the patient might miss a dose because of this or another reason. So maybe in the real world, the actual dose intensity compared to the very regimented clinical trial is not as rigid, and so doses get missed or unable to be given, and therefore the drug doesn't work as well or for as long. Which set of data are right? That's impossible to say. I think clinical trials are very important to show that the drug works to get approved. Real-world data are what I look to, right? Because real-world data reflects how I treat my patients typically and how my patients are able to be treated. And so I do look to real-world data very importantly in that regard. Even though the data aren't as rich as a clinical trial, those patients are more likely to be the patient walking into my clinic. And so I think that's, if there's a discrepancy between trial data and real-world data, it's the real-world data that I think is more applicable.